You're about to step into a mystery where science meets the unknown. Imagine zooming down through a living cell until DNA fills your view like a cosmic railway. Letters flashing past. A, C, G, T. While molecular machines glide along the tracks reading triplets as if they were tiny neon street signs. 3 letters mean add leucine. Another 3 means stop. Another 3 means start here. Every protein in every creature you have ever seen is built by obeying this same cryptic dictionary. A code that seems arbitrary at first glance, yet is shared by nearly all life on Earth. Why this particular code, with these exact assignments? And why should mitochondria or a ciliate microbe sometimes tweak it and still survive? And if you'd like to help me bring more of these mysteries to life, don't forget to like, comment, and subscribe. Tonight we're chasing one of biology's most haunting questions. How did lifeless chemistry not only invent a language, but settle, almost universally, on this one? The trail opens with the mid-20th century deciphering of the code. After DNA structure was solved, the race began to read what its letters actually said. A crucial clue was that proteins are chains of 20 standard amino acids, but DNA has only 4 bases. If the cell used pairs of bases as words, there would be 16 possibilities, too few. Triplets yield 64, enough for 20 amino acids plus punctuation. In 1961, an elegant experiment provided the first translation. A synthetic RNA made of repeated uracil caused a cell-free system to build a protein composed entirely of phenylalanine. UUU meant phenylalanine. More synthetic RNAs and ingenious logic puzzles mapped other assignments. Meanwhile, molecular anatomy revealed the cast of characters. Transfer RNAs, folded cloverleaf molecules carrying specific amino acids, and aminoacyl tRNA synthetases, the enzymes that attach each amino acid to its matching tRNA. Ribosomes, the factories, turned out to be ribozymes at heart, catalyzing peptide bonds with RNA rather than protein at their core. A biochemical Rosetta Stone had been found. Nucleic acids could speak to proteins through adapters and a decoding machine. Today we teach the genetic code as a tidy table. 64 codons grouped by their first 2 bases, wobbling at the third. But the tidy table hides a deeper strangeness. Why are hydrophobic amino acids clustered in related codons? Why do stop codons look like truncated sense codons? Why are some amino acids given 6 codons and others just 1? And above all, why is the code nearly universal, with only scattered variations in mitochondria and a few microbes? The answer matters because the code is not just vocabulary. It is the interface that makes evolution possible. Change the interface recklessly, and the whole cell stops compiling. To understand the mystery, think like an engineer and a historian at once. An engineer asks, what does this code optimize? A historian asks, what paths could have led here? The code must minimize the damage from inevitable noise, mutations in DNA, misreadings in translation, while still being flexible enough to expand. And any path to universality must explain how different lineages came to share the same assignments long before nature's branching truly branched. The classic position, the frozen accident idea, says the code is largely a contingent outcome that became locked in early because changing it would have been catastrophic. Once a population's proteins all depended on a particular decoding, any mutation that reassigns a common codon would corrupt many proteins at once. That is a strong stabilizer. But it is not the whole story, because the code's very structure looks non-random. If you permute codon assignments at random and ask, how many of those hypothetical codes would be as tolerant of errors as the real one? The answer is surprisingly few. In the standard code, a single base change often swaps an amino acid for another with similar chemical properties. Hydrophobic for hydrophobic, positively charged for positively charged, softening the phenotypic blow. This pattern suggests selection acted not only on sequences and proteins, but also on the mapping between them. 3 broad families of hypotheses try to weave contingency and optimization together. The stereochemical hypothesis argues that the mapping between triplets and amino acids is not arbitrary at all. Pieces of RNA that bind specific amino acids are enriched for the very triplets, codons or their anticodons, that represent those amino acids in the code. In other words, chemistry itself nudged the code in a particular direction. Experiments have isolated RNA aptamers that bind arginine, isoleucine, tryptophan, and others. In statistical analyses, find non-random enrichment of matching triplets at the binding sites. This is not a perfect one-to-one stamp from chemistry to code, but it hints that RNA during an early RNA world may have had a slight bias. Certain triplets like certain amino acids. If proto-ribozymes use those interactions to assemble primitive peptides, the code could have inherited those biases. The coevolution hypothesis emphasizes metabolism. In this view, the code expanded as new amino acids became available through biosynthetic pathways that start with earlier amino acids. Codons assigned to a precursor amino acid were reassigned or duplicated to serve a derivative. This idea explains some neighborhood patterns in the code. Amino acids that share pathways often share codon families, and is compatible with a chronological ordering inferred from geochemistry. Simple amino acids like glycine, alanine, aspartate, and valine were likely abundant early, while aromatic giants like tryptophan arrived late. The code learned new words when the kitchen discovered new ingredients. The error-minimization hypothesis looks like design, but is achievable by evolution. It posits that selection honed the mapping explicitly to reduce the cost of translation errors and mutations. Imagine drawing codon assignments on a graph where neighboring codons differ by one base. Then optimize the labeling to minimize the average chemical distance between amino acids connected by edges. Algorithmic searches show that the standard code performs extremely well by this metric. How could evolution perform such a global optimization? Not by searching the entire space at once, but by local moves. Duplications of tRNAs, minor anticodon mutations, tinkering with editing domains and synthetases that shuffle assignments under constraints. In communal early ecosystems, horizontal gene transfer could have spread advantageous partial mappings, while maladapted codes were outcompeted. Over time, this produces a basin of attraction, a mapping that is hard to escape and easy to adopt. Each family of hypotheses covers a different facet, and none alone explains everything. Chemistry matters. RNA can prefer certain amino acids. History matters. Biosynthesis expanded the palette. Selection matters. Error-tolerant maps outcompete brittle ones. A mature picture likely braids these together. An early RNA realm where short peptides stabilized ribozymes, and ribozymes built short peptides. Stereochemical preferences seeding a primitive mapping. Metabolic innovation and population-level selection, refining it into the error-robust, nearly universal code we know. To make the braid tangible,
Zoom in on the adapters and interpreters. Transfer RNAs carry the anticodons that read the triplets, and the acceptor stems that receive amino acids. Many identity elements that tell a synthetase, I am a tRNA for alanine, actually sit in the acceptor stem, not the anticodon. An operational RNA code that likely predates the modern anticodon-centric decoding. Meanwhile, the amino-acyl tRNA synthetases, the matchmakers, fall into two unrelated classes, each recognizing a distinct face of the tRNA. Some carry precise editing domains that hydrolyze mistakes, like a tailor who snips off the wrong color thread before it ruins the garment. These details point to a primitive world where tRNA-like molecules were selected for structural identity first. Anticodons may have been bolted on as a later refinement, when longer, more faithful translation became necessary. The codes table thus bears the imprint of an older, operational code in its foundation.
Why triplets at all? And why the wobble that softens the third position? The ribosome's geometry and the base pairing rules of RNA make a strong case that 3 base codons are the minimal units that can encode greater than or equal to 20 items with redundancy for error checking. 2 base doublet codes would have room for only 16 meanings unless you sprinkle punctuation every other step. A poor fit for noisy chemistry. The wobble hypothesis. Flexibility in base pairing at the third position. Lets the cell reduce its tRNA repertoire while keeping redundancy that cushions errors. In the standard code, changing the third base often changes nothing. Changing the first or second often swaps similar amino acids. That blocky architecture is the signature of an evolved compromise between expressive power and fault tolerance. If the code is almost universal, why isn't it exactly universal? Mitochondria read UGA, normally stop, as tryptophan. Some ciliates read stop codons as glutamine. A few bacteria reassign sense codons. And life has even added rare amino acids, number 21 and 22, selenocysteine and pyrrolysine. By repurposing stop codons when specific sequence signals are present, these exceptions are more illuminating than heretical. Small genomes under relaxed selection, like mitochondria, can afford to prune tRNAs and rewire seldom used codons. 2 mechanisms help explain such changes. In codon capture, a codon disappears from a genome due to bias mutation. Freed from meaning, it can later be reassigned safely. In ambiguous intermediates, a codon temporarily has 2 meanings. 2 tRNAs compete until one wins and the proteome adapts. Both mechanisms show that even a frozen mapping has some flex near the edges, particularly where codon usage is rare or genome composition is extreme. The very resilience of the code makes small shifts survivable in special niches. Could a different code have worked? In principle, yes. In practice, any viable complex code would have faced the same couple constraints. It may have converged on similar features. Intriguingly, when researchers compare the standard code to millions of randomly permuted codes, the standard ranks near the top for error minimization, but not at the absolute peak. That is exactly what you would expect from evolution. Very good under real constraints, not mathematically perfect. Moreover, different optimization criteria, minimizing errors, minimizing resource cost, accommodating late additions, can trade off against each other. The code sits at a compromise point where many constraints are satisfied well enough. What about the most radical speculation? That the code was designed. Science has no need for this extra assumption when chemistry and selection suffice, and the code's telltale imperfections argue against a single stroke of design. But the urge behind the speculation that the code is too good has been productive, forcing rigorous tests of optimality and inspiring models of how communal early life could collectively refine a shared protocol. One such idea proposes that before organisms diverged into neat lineages, gene exchange was rampant and codes were malleable. In that turbulent era, cells using more robust codes gained more from exchanging genes, forming a network effect. The more you share, the more you benefit from sharing a code. This collective evolution would drive convergence toward a common mapping long before descent. With modification drew clear branches. The near universality of the code may thus be a social success story written at molecular scale. Another speculative thread peers beyond Earth. If the code is a product of universal constraints, RNA chemistry preferring certain interactions, selection for error tolerance, the economics of metabolism, then alien life with nucleic acid-like polymers might evolve a code with similar architecture, if not identical assignments. Conversely, if the code is a historical accident layered atop unique Earth chemistries, then we might expect different solutions elsewhere. Even within our biosphere, synthetic biologists have begun expanding the code, adding new letters to DNA, reassigning codons, importing non-canonical amino acids, demonstrating that life can survive with altered interfaces given careful engineering. These efforts don't solve the origin question, but they stress test its answers. They show which parts of the coding apparatus are brittle and which are unexpectedly flexible. The greatest difficulty in solving the origin is that the code is both a language and a machine. To change the dictionary, you must change the interpreters, the tRNAs and synthetases, while not crashing the conversations that translation carries on with metabolism and cell division. In such a tightly coupled system, stepwise modifications must be survivable at every step. That pushes plausible scenarios toward gradual additions and local rewiring, expanding from a small alphabet of early amino acids to the modern 20, duplicating tRNAs and synthetases to create families, letting anticodon wobble extend neighborhoods, and using editing domains to police ambiguous stages. Every successful step would make the system more robust, and more robust systems would allow bolder steps. This is why the origin is so hard to reconstruct. We see only the final cathedral, not the scaffolds that once held it up. Yet, even from the finished building, we can read hints of the scaffolding. The ribosome's core is RNA, fossil testimony to an age before proteins ruled. tRNAs carry identity in their acceptor stems, an echo of an operational code that predates anticodons. Synthetases exist in two unrelated classes, suggesting independent origins, that nonetheless partition the amino acid alphabet in intriguing ways. The code's neighborhoods track both chemistry and biosynthesis. Its error-minimizing layout looks honed, but not perfectly. Its rare variations follow predictable paths where selection is weak or special signals exist. From these clues, a silhouette emerges. An RNA world that recruited peptides, a communal refinement that drove convergence, and a mapping shaped by both accident and advantage. Why should this story move us beyond curiosity? Because it reframes what life is. The genetic code is not merely a lookup table. It is a compact between matter and meaning. It allows sequences to stand for structures, and structures to fold back and maintain the language that built them. Once such a compact exists, evolution can write wonders. But before it existed, chemistry had to assemble not just molecules, but a syntax and a parser. The origin of the code is thus the moment matter began to keep consistent counsel with itself, to establish a rule that could be enforced, repaired, and inherited. It is one of the deepest beginnings we can point to without leaving science. As we surface from the molecular depths, hold on to three images. First, an early Earth where RNA fragments occasionally snagged amino acids that fit, forming tiny functional alliances. Second, a messy, communal biosphere where protocells traded parts and partial codes, and where robust mapping spread because they made exchange profitable. Third, a modern cell, a ribosome humming, tRNAs shuttling, editing enzymes snipping away mistakes, whose very efficiency hides the ancient improvisation that made it possible. The standard genetic code is not the only way it could have been, but it is an excellent way. And once it took hold, it became the foundation of our shared biological identity. We end where we began, with the code table glowing like a periodic table of life. It is simple enough to print on a mug, and profound enough to contain the history of everything that breathes. If you could rewind the tape to a planet of chemistry and watch codes crystallize again, do you think evolution would rediscover our mapping or write a different dictionary? And what, in your view, would be the strongest forces nudging it one way or the other?